everyone. So today I am going to discuss about the antigen antibody interactions as I mentioned in the previous video. So let's start it with the precipitation reaction. So precipitation reaction occurs between an antigen and an antibody. So that is why it is under the section of antigen antibody interactions. So let's look what is it. So an antigen. So whenever an antigen reacts with an antibody, and the criteria is that for a successful precipitation reaction, the antibodies and the antigen must fulfill three criteria. One is that the antigen must be polyvalent or bivalent. Polyvalency of antigen means multiple epitopes should be present on the surface of the antigen so that multiple antibodies can bind with those epitopes. So this is the valency of an antigen. Antigen must provide surface for the binding of antigen. And antibodies, the second criteria is that the antibody must also be bivalent in nature. Antigen must also be bivalent. So what is bivalent antibody? The antibody must be able to bind with two antigens simultaneously. So this is a valency of an antibody. So whenever these two criteria are fulfilled, a structure like this forms, where this is an antigen, these are the antigens, these structures are the antigens and these are the antibodies. So one antigen is bound simultaneously by two antibodies. One antigen is bound simultaneously by two antibodies. So whenever this scenario comes, the thing is that a visible precipitate forms on the medium, because a large chain-like structure forms, a visible clumping occurs because every antigen molecules are interconnected by the help of antibodies. Antibodies and the antigens form such a structure, such an interconnected network, that a visible precipitate forms on the medium. So this is why the name precipitation reaction is termed. So this is about the precipitation reaction. Now we will see that three cases. The first one is that when antigen is in excess amount. So what will happen if antigen is excess? Antigen is in excess amount. So there are innumerable antigens on the medium floating, and there are only few antibodies. Only few antibodies are there, but antigens are in excess amount. So that interconnected network cannot form, that I have shown in the first picture, that an interconnected network of antigen and antibody will form, that will ultimately give rise to the precipitate. So in this case what happens, only few antibodies are present but antigen molecules are in excess amount. So in this case precipitation reaction is not possible, precipitate will not form. The second case is that antibody is in excess amount. So antibody is in excess amount but antigen is very few. So in this case also precipitate cannot form because antigen is very few in number but antibodies are more. So in these two cases, in case 1 and case 2, where antigen and antibody are, any one of them are in excess, in either case precipitate cannot form. Only if the antigen and antibody are present in a certain proportion, in a specific proportion, only in that case precipitate will form. So if the structure is like this, only in this case the precipitate will form. An ideal case is that antigen is present in slightly excess amount than the antibody. Antibody must be present in slightly lower amount than the antigen. In that case only the antigen antibody, the precipitate will form. Coming to the test based on the precipitation reaction. The name of the test is radial immunodiffusion. So we know what is a diffusion. Diffusion means free movement of solute molecules from higher concentration to lower concentration. So the solute molecules are moving from the position on the medium where its concentration is higher to the medium where the concentration is lower. So the theory is basically, the principle is all the same, that diffusion, how diffusion occurs, we know that immunodiffusion means it is basically an immunological technique. So it is immunodiffusions. The immune molecules are diffusing in the medium. That is why immunodiffusion. And why it is called radial immunodiffusion? Because the diffusion is taking place radially. We will see that the whenever we as we proceed, we will see that the migration will occur along a radius. A circular radius will form. That is why the name of the test is radial immunodiffusion. We come to the we measure the result by measuring the radius of the structure, the radius of the ring that is formed by the immunodiffusion technique. So that is why the name of the test is radial immunodiffusion. Now let's look the details of the reactions, how the reaction happens. The first thing we need for the reaction, I am giving the step by step reaction. So the first thing that we need is that a medium. We should prepare a medium first. So the medium in this case is an agar medium. Agar medium. So agar is a gel. When the agar solidifies, it forms a gel-like structure. So we should prepare an agar solution and heat it to solubilize the agar. And now after heating, first we should heat the agar. Heat and solubilize the agar. Next, we should cool it slightly. Then we get a solution like this. Agar solution like this we get, and now in this solution we add antibodies. So we add antibodies in the solution. Now what happens? This agar solution is poured in a plate. So we take a Petri plate, and in that Petri plate we pour the agar containing the antibody solution. So at this point, this agar medium contains the Petri plate contains agar containing a solution of antibodies. So the antibody solution is mixed with the agar and the plate is allowed to solidify. Now, after the solidification of the Petri plate, we dig small wells on the Petri plate. So if we look from the above, we will find the plate will look like this. So this is a Petri plate, and these are the Petri plate with wells containing in the Petri plate. So these wells are then filled up with antigen solution. So we add antigen solution in the wells. We add antigen solution in these wells, and now this plate is allowed is kept for some time for the reaction to happen. So what will happen? These wells contain high concentration of antigen, right? This antigen, the concentration of antigen in this well is high. So it will try to diffuse out into the medium. It will diffuse into the medium and migrate along the medium. And what is present in the medium? This agar medium contains agar plus antibody. Agar plus antibody is present in the medium, and the antigen diffuses out from the well through the medium. So whenever in that position of the plate, in that region of the Petri plate where the concentration of the antigen and the antibody matches that particular proportion for the precipitation reaction to occur, a visible precipitate ring will form. So a visible precipitate like this will form on the surrounding the well, right? A visible precipitate ring will form surrounding the well. So this is why the name of the test is radial immunodiffusion, because if we measure the radius of this precipitate ring, if we measure the radius of this precipitate ring, we can quantify the amount of antigen present in the solution. So the radial immunodiffusion is basically a technique to find the unknown concentration of either antigen or antibody. So if we unknown concentration of antigen can be determined by this technique. And if the opposite case, if the an agar solution is prepared instead of antibody, the agar is mixed with a solution of antigen, and we put the unknown concentration of antibodies over these wells. Within these wells, we keep antibodies. So the similar reaction will happen. The region on the plate where the concentration of the antigen and the antibody matches that particular ratio, precipitate ring will form. And if we measure that ring, we can find out the unknown concentration of antigen or the antibody by this method. So this is all about the test radial immunodiffusion. It is based on the principle of precipitation reaction.